Okay, let's get started. Good morning, good afternoon, or good evening to everyone. Thank you for joining today's webinar. My name is Jim Dobrovolsky, and I am the National Program Leader with the National Institute for Food and Agriculture, or NIFA, at the United States Department of Agriculture, and I'll be your moderator for today's webinar. The webinar is titled Environmental Health and Safety Implications of Metal Nanoparticles in Aquatic Environments. The National Nanotechnology Initiative, or NNI, has various mechanisms to foster and support interagency collaboration and community engagement in areas of national priority. This webinar is supported by the NNI's Water Nanotechnologies Community of Interest, which aims to support water sustainability through nanotechnology. The webinar will explore the environmental, health, and safety implications of metal nanoparticles in aquatic environments. The focus will be on copper and copper oxide, nickel, and silver nanoparticles. Each expert panelist will provide an overview on the current state of knowledge for a particular metal nanoparticle. They will discuss methods used for identifying, measuring, and assessing its potential effects in aquatic environments. You can type your questions into the Q&A box on the bottom of your screen. We'll try to get through as many questions as we can, and I look forward to a lively conversation. The chat function will not be available, so just put your questions in the Q&A, please, and we will hold the questions until the end of the panel discussion. So let's briefly introduce our speakers for today. Our first speaker is Dr. Stacy Harper. She is a professor with a 50-50 split appointment with the Department of Environmental and Molecular Toxicology and the School of Environmental Engineering at Oregon State University. So Stacy, the floor is yours. Take it away.
Okay, great. Thank you for the introduction. Able to see my screen now?
Yes.
Okay, all right, great, thank you. So my focus for today is going to be on copper and copper oxide nanoparticles. So I wanted to start with, where are all these copper nanoparticles coming from? So copper has a bunch of features about it that allows for it to redox cycle, allows it to engage in Fenton-like reaction, can spontaneously generate reactive oxygen species. And a lot of these properties can be leveraged into materials for things such as gas sensors, batteries, metallic coatings, catalysts, and rocket propellants, and antimicrobials, antifungals. But there are numerous applications in agriculture specifically. So if we look at this diagram, it shows us, we can intentionally make these copper nanoparticles and infuse them in everyday products. But there's also unintentional sources of copper-based nanoparticles. In the ag sector, micronutrient supply, insect pest management, fertilizers, nanoherbicides, and like I said, fungicides are all widely used applications for copper nanoparticles. And these copper nanoparticles then make their way into the ecosystem, right? If you're applying them to an ag field, any runoff from that field is going to go to an aquatic environment. And so we're going to focus a little bit more on the aquatic environment. So toxicity varies widely depending on the species that you use, but also the test procedures that you use. So changes in the water chemistry, exposure conditions, when you do an exposure, these are all going to have an impact on how toxic copper nanoparticles are. And if you can see from this review from a while back, that really compiled information across a bunch of different aquatic species and looked at the relative toxicity. And you can see down here, algae and Daphnia were rather sensitive to the copper nanoparticles. And these were copper oxide nanoparticles in this review. The EC50s, you'll note for algae and for Daphnia, have a very, very wide range, and that's for a lot of other organisms too. And this is due to this variability in exposure conditions. And when we talk about fish, there's a recent review in 2020 that came out looking at toxicity in a variety of different fish species. And some of the common things that they come across are causes liver dysfunction, kidney necrosis, olfaction impairment, and you can imagine for fish that need to hone to get up to their reproductive spots like salmon, olfaction impairment can have long-term ecological consequences for those organisms. They've found organ damage to the brain, to gonads, heart, impacts on the mucosa, gills, any of the epithelial type linings, and altered plasma osmolarity, which is likely due to decreased sodium potassium ATPases at work. So we had the question of, all these exposures are done in single organisms, but we know that organisms don't live independently in the environment. And so we established these small scale microcosms where we can do our exposures of algae, bacteria, Daphnia, and zebrafish, and we optimize how many organisms we could have in this, 50 ml flask to do a small scale kind of microcosm. And we assess the impacts in these organisms to copper and copper oxide nanoparticles, and then of copper salts as a relative comparison. And so what we see is the copper nanoparticles themselves, both the copper and the copper oxide, the primary particle size was about 50 nanometers, but you can see that they do agglomerate. Here's hydrodynamic diameter, but the agglomerates are relatively similar. And then these are just the zeta potential. And it should be noted that the outside of the copper nanoparticles do have a 2 nanometer oxidized outer shell. So the outermost surface is actually identical. But what we see is differential uptake of these particles. The copper salts are not taken up at all. But you can see here the copper oxide, and these are, the copper found in Daphnia. And you can see the copper oxide nanoparticles at the higher concentrations are readily taken up by the Daphnia. And so what does this mean consequentially for them? When they're in these small scale microcosms, what we see is that copper here is the dark one. It's always the lowest line on the graphs. Copper significantly inhibited algal growth and Daphnia survival and zebrafish hatching. But copper chloride and copper oxide nanoparticles only impacted algal growth, so and in a very similar fashion to the copper. So this means that the copper nanoparticles potentially have mechanisms of toxicity that are different than the ionic form or the oxidized form. So there's some additional data gaps that we noted when doing these studies that we wanted to move to a more chronic exposure, because we know Daphnia and other aquatic organisms are not likely exposed at a single point in time. They're exposed chronically. And we believe that trophic transfer through aquatic food chains had not been well studied. And so we wanted to target some of that, answer some of those questions about potential impacts of direct exposure versus impacts of indirect exposure from a food source being contaminated. And we know that predators live in ecosystems that can contain contaminated foods as well as contaminants. We wanted to tease this apart a little bit. And we predicted that they might have differential effects if they're exposed through their prey versus just exposed in the water column. So we set up, based on a couple of hypotheses that we had, that the delivery might alter the toxicity of the materials. We had a direct waterborne exposure where we just fed algae and copper oxide nanoparticles to Daphnia at the same time. And we had an indirect feeding exposure where we pre-incubated the copper with the algae and let that incubate for a while. and then centrifuged them and collected them. And our exposures were basically a feeding of just algae alone. And then we had a high concentration where we did this incubated algae here. And then we did a low one where we did half and half just regular algae, non-exposed, and half of the incubated algae. And we did these over 14 days, and we renewed the water every 48 hours. And we looked at copper uptake into the Daphnia themselves, into neonates that they produced during their exposure, and the molted carapaces. We looked at things like body length and mortality and reproductive success. Identifying the copper oxide nanoparticles, we have a Cytoviva system for hyperspectral imaging analysis, where you can basically take a scan in dark field microscopy, and the metal nanoparticles in particular are really, really bright and distinctive. And so we can train the library to identify the copper nanoparticles. So this is an example of copper alone. We have the copper oxide mixed with the algae. You can see if you probe the area with the algae, it gives you a very different signature than with the copper nanoparticles, and you can see them visually. What we found is over time, copper concentrations in Daphnia continually increase for those that were in the direct exposure. So these are these two bottom brown lines, the low and the high direct exposure. But in feeding exposure, we see that there's kind of a plateau here at 9 days where they didn't really take up any more of the copper. And the feeding exposure also showed a more rapid increase of internalized copper. And I noted here at the bottom that the dissolved copper that was in the system was very, very low. Okay, so what are some of the impacts that we saw on Daphnia? This is survival. This next one is the number of neonates, and then the number of broods that were produced. And there was no significant mortality found in the feeding exposure, seen right here. But we did see a significant mortality after 14 days in these that had the direct exposure. We also see a decrease in the number of broods and the number of neonates that were produced during that 14-day exposure. And there was a lower number of broods observed in the direct exposure compared to control. So where did this copper go? So we have on the left here, copper molted in the carapace, and then we have a copper that was actually found in the neonates as we pulled them out. And the feeding exposure resulted in more copper found in the carapace than in the direct exposure. So that was interesting. But even more interesting was here in the feeding exposure, where we see copper being transferred basically to the neonates. So copper is ingested and then winds up in the neonates. So here's a result summary for you. In the indirect feeding, where we had the copper was bound to the algae, we had less toxicity in the adults and less uptake, which is probably correlated to the lesser toxicity. We saw large amounts of copper in the molts, in the molted carapaces of the adult Daphnia, but also in the neonates. And then here in the direct water, where we did kind of a competitive feeding, we see increased toxicity and increased uptake in the adult Daphnia. We see less in the molts and less in the neonates. So what this means is the high concentration of copper in the carapaces and copper found in the neonates from the feeding exposures mean that Daphnia may be able to regulate their internal copper through molting, but also through maternal transfer to the offspring, which is kind of unique. Can be transferred, copper oxide can be transferred through the food chain and impact organism health in a way that is dependent on the delivery scenarios. We saw when it was pre-incubated with the algae, we saw a different response than when we just did the direct exposure. So I would say, as far as needs go, still, trying to move towards more environmentally relevant testing scenarios where we can use, multiple delivery scenarios or multiple exposure routes that they would typically be seeing in the ecosystem. The further development of rapid multi-species assessments. So we miniaturized this assay because number one, we don't have enough nanomaterials to do a full ASTM study, nor would we want to generate that much waste. And so I think, paring down and making small scale microcosms where we can ask community level impacts is really important. And we're going to need to be testing all of the new novel formulations of copper-based nanopesticides, nanoherbicides, nanofertilizers, and all of the like. So I will stop sharing here and turn it over to our next presenter. Oh, thank you.
All right, thanks, Stacy. Appreciate that. This is Jim Dobrovolsky again. Our next speaker is Dr. Custodio Muyanga. He's an environmental health scientist at the Agency for Toxic Substances and Disease Registry, the ATSDR, the National Center for Environmental Health, the NCEH, at the Centers for Disease Control and Prevention, the CDC. Custodio, the floor is yours.
Thank you.
Your slides are
Can
up.
you hear me?
Yes, sir.
Well, thank you for the opportunity. I'm going to briefly discuss preliminary results of a review of potential human exposures to nickel, both metallic and nickel compounds, nanoparticles from aquatic environments. I'm going to also touch a little bit on the health effects and give some public health options that we can use to reduce exposures. Just to make it clear, I'm going to present these discussion points, and I'm not going to present any additional slides because I'm still working on the work, but I will be happy to entertain any question associated with engineered and incidental nickel nanoparticles. Briefly, I will discuss about the synthesis, the characteristics, a little bit on the application. And when we go to the exposure, I want to discuss a little bit about the life cycle and be able to discuss about the fate and transport. They are great two examples of oral exposure. So this is, we are talking about aquatic environment and focusing on people, then will be water. And so I will give some examples of data in the waste water treatment plants and landfills. At the end, I will discuss some health effects and some data needs at the end and give some options on managing exposures. Just to start in the first point, nickel nanoparticles nowadays are being produced for many uses. And the main process that is being used is through the reduction and oxidation reactions. Most of the time they will use aqueous solutions, but they can use other type of solutions depending on the uses. The typical size ranges, which mostly are controlled, are somewhere from 10 to 100 nanometers with varying specific surface areas. And this again has to do with the type of uses, which is these nanoparticles are popularly used in the mechanical, optical, electric, and biosensing, catalytic converter, especially in the automobile industry, the list is huge, you know, microelectronics in the packaging industry. So this is kind of important to other group that we discussed on the microplastic, because you have these nano nickels in the packaging and the propellant additives, but also you can use in the solar cells and the water purification systems. When you look in terms of the market annual growth, the Fortune business inside estimate an annual market annual growth of about 10%. Just to see, before COVID was somewhere around 22%. So it means that there were a type of decrease, and hoping that this will pick up again up to 2029. So those are kind of the estimates. Then when we want to start thinking about exposure and think about health risks, I think the best approach is to go to the life cycle assessment. I remember we discussed in meetings previously. So the life cycle assessments allow us to get the opportunity from the production, use, disposal, but also the transport and the fate and the key processes within the process. You know, just giving an example, great compartments in the production I already discussed, once they are being used, at some point they will end up in the waste treatment, wastewater treatment plants. They will end up, for example, in the waste incineration plants. They will end up, for example, in the landfills. You know, this is in the process of disposal. And then as you start looking on there, either from the production, most of the time, the workers are very well controlled, because these processes, most of them are closed processes. But when you go to these disposal parts, you have so many influences of other factors. And these may end up in somewhere in the surface waters. And then from the surface waters, we may get exposed. The good news is most of the waste treatment plant technologies that we have in the United States and many developed countries are very efficient. So we have a removal of these metal-based nanoparticles in general of about 90%. It is true that for nickel, nanoparticle actually is a little bit down around 78%. And then these will be going through the process. Then they were removed, and then they will go to the surface water streams. But then we also have the drinking water systems that are very, very efficient. You know, now with the advanced technologies, they will also be able to remove some. But you know, it is reasonable to have some concerns, especially in the landfills through the leachate. If you are familiar with the leachate, has a good amount of metals, which they will come from all different processes, incinerations and all that. And these nanoparticles are bound in the colloids. And we all may be familiar with the colloid science. It is a long time, but then you find them. They are a very good amount of studies that they were able to detect and quantify these nanoparticles. One of the efficient way to be able to detected. The use of ion exchange resin has been very successful in first removing the dissolved form of nanoparticles, including, so the metal-based nanoparticles, including nickel. Usual nickel is ranked in the range, you will find the majority of, for example, magnesium, titanium, iron, copper has kind of the major that will go first on these in 90% first, and then you will have a nickel, and with others that will come a little bit before. Once you have removed those that are dissolved, then another instrument that is used initially started as a single particle inductive coupled plasma mass spectrometry. But that process is very complicated because you go through only one by one metal. And then now they are a little bit more sophisticated, where you can have multiple and be able to collect efficiently. So these are kind of good examples, but then you can be able to see as you continue collecting in a transmission electron microscopy. That is kind of a basics. Just to give you some ideas in terms of some concentrations in general, for example, in the landfill leachate, you will find the nickel concentrations somewhere between 10, maybe less than 100, to tell you, less than 100 nanometers per liter. But you can find the other metals at high concentrations than that. But nickel will be somewhere less than 100 nanograms per liter. And these can be either in the beginning. You can also find in the waste treatment plants. In different stages of the waste treatment plant, you may find there are good amount of research that they have done in different parts. So as I was saying, you can be able to visualize this, and using the cascade filtration process. It means that you will have a microfiltrate, you will have a ultrafiltrate, and then you can use instruments, as I said, transmission electron microscopy is a great example to be able to see, but also the energy dispersion spectrometry has been used successfully. So you have that in the municipal solid waste landfills, both the active and the closed one. Usually the active landfills, they have a higher levels relatively compared to the closed one. And that has to do with the biochemical processes that are associated with the occurrence and suspension of these. For example, if you have ions of chlorine and sulfur, then you have a good dynamics there. So in terms of exposure, or for human health exposure, now we are focusing on the oral exposure. Definitely there is some data in the oral exposure. Mostly is in inhalation and some of the dermal. On these oral exposures, nickel nanoparticles, most of the time we may get them through drinking water or food chain. Now there are some attempts with the advanced technologies to make sure that And these will be the best, have better technologies to eliminate those concentrations. In terms of toxicity and human health effects, there is also a very good amount of studies, both in vitro or in vivo studies. And very briefly, they all are consistent on the nano and the nano nickel. Both the metal and the compounds induce DNA damage through the direct DNA binding and the intracellular reactive oxygen species stimulation. There are effects in various cells, but also in animals, and some human studies. Most of them are acute studies, the one that will define, for example, the lethal doses. If I were to present them a wish list, will be, for example, a robust epidemiological study, a robust toxicological study, which used the main experimental animal laboratories. So the robust studies that we have in nickel, which only focus on the nickel at the micro level, they were done in 1996 by the NTP. So then it is good time to update that. But we know, that nickel in general, it is known to be human carcinogens. The compounds and the metallic one is reasonably anticipated to be a human carcinogen. So just a little exposure may have some impacts. Looking on the eye as a public health professional, we are more concerned of people that live nearby, for example, landfills, wastewater treatment plants, these waste incineration plants, and the water streams may have some contamination. And another way, a way to bring that up, will be to educate the population. And you can do that by bringing, for example, a web-based information where because the processes are all the same, and all these are from the organic waste materials. Then you can provide that information. It means that, for example, best practices in landfillings will be useful. The sustainable compostings will also be useful to make sure that those nanoparticles are eliminated throughout the process. That is the discussion, and I will be happy to sustain more as we move forward. Thank you.
Thank you, Custodio. And now we're going to have our last speaker. And that is Dr. Olga Susko. She is an associate professor in the Department of Plant and Soil Sciences at the University of Kentucky. So Olga, take it away, and your slides are up.
Thank you, Jim, for the introduction. And let me just put my laser pointer. Okay, I'll be talking about silver nanoparticles, and you already probably know that silver is one of the most widespread of the products on the market, on global market, that contains silver nanomaterials. And the predicted basically global market for silver by 2027 is estimated at 5.5 billion. And it's based on the growth rate, annual growth rate 14.5%. So silver nanoparticles are being used in all different fields, the application, but like 75% of the products that are being developed are in the health due to antimicrobial, mainly properties of silver nanoparticles, but also silver nanoparticles have unique optimal electronic catalytic properties and all obviously find more applications in other areas as well, including like human gardens expanding. Okay. So Dr. Miyank already was mentioning about life cycle when he was talking about nickel, right, nanoparticles. So how do nanoparticles in my case, it's silver, enter the environment, the aquatic environment? So if you look at those blue lines, there are several ways for them to reach aquatic environments. So for example, after use, nanomaterials enter wastewater, and after wastewater, they partition to sludge and apply to agricultural soils as fertilizer. And during surface runoff, they are reaching aquatic environment. Another way is through industrial discharges during production. So in both cases, they're kind of like entering through wastewater. What I want to emphasize is that the particles that are incorporated in the product, let's call them pristine silver nanoparticles. And the particles that reach aquatic environment are quite different due to different transformations. One of the most common transformations is in the presence of oxygen. They're oxidizing and slowly releasing silver ions. And silver ions, as you probably know already, are highly toxic. However, for example, when they enter wastewater, right, the environment that is very rich with sulfur, silver nanoparticles will be sulfidized, often completely sulfidized or partially sulfidized. And then their solubility will be decreased a lot. So now during surface runoff or direct discharge of wastewater into aquatic environment, there is also present natural organic matter like humic acid or fulvic acid, right? How does that affect now stability of those sulfidized nanoparticles? So, and this is just kind of a simple view, right? Talking about sulfidized nanoparticles reaching environment. Now there is more stuff that can basically attach and adsorb to the surface of those sulfidized or partially sulfidized silver nanoparticles and form ecocorona. So those are environmental factors. And among them is not just natural organic matter, but also extracellular polymeric substances that are secreted, for example, by microorganisms, right? There is proteins that are secreted by fish by releasing bodily fluids, for example, or through mucus secretions, through bivalves. And there are also discharge of different surfactants. So all of those can be adsorbed to particles, and eventually what the organisms see and being exposed to are totally different from pristine particles. So the properties and toxicity directly related, and actually the transformation of those nanoparticles are directly affect the toxicity and bioaccumulation of those particles in the organisms. So one of the main ones when we are talking with pristine, and a lot of study was done with pristine silver nanoparticles, it's solubility. So they're dissolving and releasing silver ions. Another thing that during environmental transformation, there is a change in their surface modifications. Again, it changes their properties and hence toxicities. So this is just species sensitivity distribution specifically for bare non-coated silver nanoparticles. It's kind of statistical approach tool, and it's based on separate 10 studies for LC50, so 50% in mortality. And the distribution is fitted with 95% confidence interval for all those 10 species. As you can see, the most sensitive species, and this is a tool that risk assessment can be used to establish environmentally safe kind of concentration in the surface water. And on the bottom of this, which is the most sensitive species, is actually Daphnia. And then we can see that there are other crustaceans and fish, such as Modaca. Then we have algae, fathead minnow, and zebrafish. Right. So based just on those baroparticles, this is what species sensitivity distribution looks like. However, if you look at species sensitivity distribution curves for particles that are, for example, coated, or for silver ions alone, then it's even more steep and there is even more toxicity. So the question is, when those particles are transformed, would they be dissolving or not? So this is just results of some study on zebrafish cells. And just to demonstrate and illustrate the toxicity of the silver nanoparticles, when they're being taken into organism and being absorbed into the cells, often they can dissolve before entering the cells, and they enter cells as silver ions, or they can be taken in through endocytosis and enter cells as silver nanoparticles. However, if they're going through endocytosis, eventually they will reach lysosome. Lysosomes are a very acidic environment. The silver nanoparticles will be dissolved and silver ions will be released. Hence, as a result, there is increase in generation of reactive oxygen species. It can lead to apoptosis. It can affect mitochondria performance. It can also cause some genotoxic effects. And in this diagram, you can actually see also in zebrafish cells the different basically toxic effects in terms of different size of particles and different concentrations of particles. And at the end, right here on the bottom, you can see those toxic effects kind of separated for silver nitrate. And if you compare a silver nitrate distribution of those toxic effects and toxic effects in response to the smallest nanoparticles, in this case is 10 nanometer silver nanoparticles, then there is a lot of similarities, right? A huge proportion is being taken by necrosis. There is hardly any healthy cells left, and as we increase concentrations, those healthy cells completely eliminated. However, with increased size, the distribution is different. We still see a lot of toxicity, but it's different from silver nitrate. So now just a couple examples. For example, if we look at pristine versus transfer silver nanoparticles. In this study, the silver nanoparticles were called wastewater-borne silver nanoparticles. Basically, pristine silver nanoparticles are introduced through pilot sewage treatment plant, and then that wastewater was used for exposure of daphnids in this case. And if we compare it with pristine versus wastewater-borne silver nanoparticles, there is definitely reduced reproduction over multiple generations in response to pristine. However, we don't see those effects in response to transform. And those are likely just sulfidized silver nanoparticles. This is another study in rainbow trout, the same kind of design. Again, even though there is a decrease in bioavailability of the wastewater-borne silver nanoparticles, and there is no effect on growth, there is still effect on protein synthesis, lipid hyperhydrates. Now, I was talking about more sulfidized transformed particles, right? What about humic acid? Because when silver nanoparticles in the environment and they get coated with humic acid, what happens? So basically this study demonstrates in adult zebrafish that if you look at the distribution of the particles among different organs, it doesn't change whether they are coated with humic acid, whether they just pristine. However, it does decrease their viability if they are pristine, and it's actually increased viability by 50% when they are coated with humic acid. Also, there is less uptake a little bit, not by huge percent, but some. So I know this is C. elegans, and it's the organism I'm working with. It's a model organism, and because it's sometimes found in sediments, and it also lives in soil poor water, it's being used as model organism for aquatic toxicity testing and for terrestrial toxicity testing. So those exposures were done, aquatic exposures, and again, we were working with pristine nanoparticles and with sulfidized transformed nanoparticles. As you can see, the toxicity basically in response to silver ions right here is pretty steep at 5 micrograms per liter. Right above, we start seeing some effects in toxicity in terms of reproduction. However, the toxicity is decreasing as soon as we switch to pristine, and it's definitely even less toxic when they are sulfidized. If we look in terms of multi-generational toxicity of also pristine silver ions and sulfidized silver nanoparticles over 10 generations. So just kind of walk you through what we see here is the concentration that is causing 50% decrease in reproduction. And you can see as soon as second generation, that concentration is dropping significantly down, and it's consistently persisting up to F10, meaning that you need to have much smaller concentration to cause 50% decrease in reproduction. Exactly the same pattern is observed in response to pristine nanoparticles. And in response to sulfidized, we don't see these effects until we reach generation F10. To make matter more complicated, the salinity can increase actually toxicity of silver nanoparticles. Why? Because when we have fresh water and there is just a little bit of chloride present in the water, then the silver will form pretty really stable, not dissolvable, basically, compounds. However, if there is increase in salinity, then there is formation of soluble silver chloride complexes. And actually, there is increase in toxicity in this case, in this study in Medaka embryos. Okay, at the end, what I want to summarize that those effects, environmental and health effects, are very complicated. It all depends on environmental factors, on biology of organism, and obviously properties. So basically the question still remains, how can we reduce or minimize the toxic effects from silver in the environment and human health? And one of the potential strategies where we need to take, for example, the risk assessment, life cycle assessment, and functionality, and finally cost all into account and design more safe, basically, not nanomaterials, but actually the products. With that, I stop.
Okay, thank you, Olga. That was wonderful. Appreciate that. Thank you, speakers. And what I'd like to do now is go to the Q&A, and we'll answer some questions that are listed there. And so question number one, is there any computational model to evaluate the toxicity of nanoparticles? And who wants to tackle that one?
I can go ahead. There might be some others that Olga and Custodio could think of. I would say in the US, the Center for Environmental Implications of Nanotechnology, one that was hosted in Duke and one that was hosted at UCLA, had nice databases where you could query, for nanomaterial properties. UCLA also has what's called MEND Nano, multimedia environmental distribution. So you can look on the exposure side of risk. And then in Europe, there are a lot of computational models, and they'll also form partnerships with US researchers through different type of work packages. eNanoMapper is one of the premier ones. Nano Commons, let's see, what are some of the other ones? Olga? Those are some of them. So they are available, yes.
I know you already mentioned the ones that I could remember. The NanoMapper.
Oh,
and
NanoSolvit. NanoSolvit is another one of the European Union's collective attempts at getting together not just all of the data, but the tools and analytical support for analyzing that data.
Great, thanks very much. Our second question is to you, Stacy.
Mhm.
Based on your experience, whether metal nanoparticles or metal oxide nanoparticles, which one is more toxic? And is it based on its size or morphology or surface charge? And is there any trend of toxicity based on the presence of oxide?
Don't know that you could generalize that. So the solubility of the metal or metal oxide would certainly play a role in it, particularly when the ions are the toxic component. And I would say you probably couldn't make a general rule about a metal versus a metal oxide. It really depends on the solubility of that particular metal.
Great, thank you.
Mhm.
So Dr. Muyanga, is there enough information on behavior and fate of nickel nanoparticles in the environment? i.e., do they agglomerate to become less concerning from the size point of view, or persist in their life cycle as nanoparticles?
The first question is, yes, there is a good information. As I said, as an environmental health scientist, I would like to see a robust epidemiological and toxicity studies that will allow us to see a good dose response. The majority of the information that exists is in vitro studies. But they are actually very good data that has been empirically done. And I can share this information if you want. You can follow up. Mostly in the municipal solid waste landfills, in the wastewater treatment plants. And most of the data that I've seen have been done in Europe. In terms of behavior, what is happening most of the time, it depends on the physical chemical properties. If you're familiar with nickel, has a multivalent, and that's why you have the reduction and the oxidation reactions in that processes. So yes, they have been found binded within the colloids and has in nanoparticles themselves, but also there are some agglomeration. Actually, maybe I can, there are some agglomerations, but there are also other processes like dissolutions, depending on the environmental conditions that you have within the streams.
Thank you, Custodio, very much. So, Stacy, did the algae in your nano copper toxicity study include cyanobacteria? As you know, harmful cyanobacteria blooms are becoming both a scientific and a public health challenge.
Good question. No, we used Chlamydomonas for these particular studies. I don't know if the person asking was going for something like treating cyanobacteria with copper nanoparticles, but probably not a good idea for all of the other critters that live there. Don't know that it would specifically target the blue-green algae.
Great, thanks.
Mhm.
So this is for Olga. In looking at silver particle toxicity and transformation, it seems the sulfur coating transformation, in addition to oxidizing to AgO, decreases toxicity. Is this solely because of a decrease in silver ions or other reasons with surface chemistry? And could this mean engineered silver nanoparticles should have an initial treatment coating process to mitigate risks? What do you think, Olga?
Okay, there is still part of the question that I'm still reading. Thank you, Austin, for the questions. So yes, you're right that sulfidation, yeah, I think the decrease in toxicity is really due to solubility. So there is huge decrease. It's very stable particles, but they're completely sulfidized. The problem is if they're partially oxidized, right? So they still can release ions. Well, once they enter wastewater, they are pretty much starting sulfidizing. The problem is if they reach surface waters, right, and enter aquatic environments, then there is interaction with other organisms. Will they continue being that stable or not? You know, if forming that ecocorona will actually destabilize them, then we have release of silver ions. And you're looking about initial coating process. So no, I would think that right now the water treatment process is really targeting for filtration and removing of silver particles, nanoparticles from the water. And those methods are pretty successful. However, smaller size and smaller ones, the ones that actually prone more to dissolution, are the ones that kind of like goes through. So I think maybe on improving of the removal methods would be like the target for treatment. But the coating and changes of the coating, maybe that makes sense when you actually designing the products and incorporating. If silver is impossible to replace with anything else, then be at least to make it as stable as possible through surface modifications.
Thanks, Olga. So we have time for about one more question. And so we're going to pick one. How do we practically set protection levels? For example, the MRL or the RFD, or safe use practices that consider the wide variability and toxicity through FATE processes. Who wants to take that one on?
So I can start. So this is one of the challenges that so far we have. You know, in my preliminary analysis, I was able to check in terms of dose response, potentially to develop any toxicity. Most of the studies, they really become short on that perspective. There are some acute studies that, you know, at least you will be able to determine lethal doses. But then I think at least for nickel, you know, what I have seen for nickel, and there are some, for example, in the inhalation route of exposure, there are some preliminary studies that go a little bit to like 14 days. I remember there is one study that I saw that go for 28 days. But if you want to do a good toxicity assessment, you want at least an exposure or dosing that go to 90 days start at least. And then if you have good opportunities, then you want a chronic study, a 2 years experimental laboratory study that will measure both the exposure and the effects. And you want also a multi-generation reproductive studies. We don't have that yet. So
Okay,
my
very good.
recommendation is will be nice to work on this, especially for our fellows that they have those laboratories. Thank you.
Okay, thanks, Custodio. We are out of time. Just one final comment before we adjourn for the day. I hope you'll join us for future NNI public webinars. More information and recordings of all of the NNI public webinars can be found on nano.gov. You can follow the NNI on Twitter at NNI Nano News and on LinkedIn at the National Nanotechnology Initiative. Thanks very much for coming and attending and your attention. And we'll see you next time.
Thanks.